biosensors could be very useful if we want to start doing more on-site testing, such as field testing, or potentially in more obscure locations. And so one of the methods that we really like to employ, of course, are fluorescent biosensors, where we very simply develop a complementary probe to the aptamer, and we label the probe and the aptamer with FRET pairs. They could be fluorescence quencher, which is what I'm showing here. And so again, the light is a bit difficult, but basically when the probe is displaced by target binding, this allows for the aptamer to fluoresce, so that you can detect this quite easily. But actually the motivation for this project is my interest in looking at sort of aptamer folding and dynamics. And I won't get to talk about what I really want to do, which is single molecule biosensing with aptamers. But this project was really inspired by my dream to look at aptamer folding dynamics and how they might be quite different if we look at them as a single molecule, right? And the thought there is that in bulk, if there's heterogeneous behavior, we can't detect it. So you really have to look at the single molecule. But the Shostak lab and coworkers in 2009 actually showed that some, or in this case, GTP, an in vitro selected aptamer, displayed homogeneous behavior. But we know things like riboswitches go through different pathways to find their targets. So I think that now that we're getting into capture selects and complex biosensing, I do think that there's some interest in further looking at this type of behavior. And so again, I'm not going to say anything more about the single molecule stuff. Hopefully next year we have some data, but this project really stemmed from having a starting model aptamer biosensor that we could use for the single molecule biosensing. And so ochratoxinA, you've already heard it here today. It's a really amazing aptamer that I think there are probably hundreds of papers describing biosensors for ochratoxin. And so we looked in the literature and we found that there were many, many, many papers that just did this simple strategy of using a complementary probe to the aptamer and using FRET pairs and fluorescence quenching to detect a signal. And so we thought, okay, let's test a whole bunch of them and see which one works best for our single molecule studies. And of course, this is where we got stuck right away. And so when we compared these published probes, we found that when you add the target, often the fluorescence would increase because the quenching complementary probe was kicked off the aptamer, but a lot of them didn't work. This is really discouraging because I think being an aptamerist for a long time, we're always kind of panicked when an aptamer doesn't work. You know, we don't want our field to be sort of disgraced when we can't reproduce something. And so we decided to look at it a little bit more carefully, and it's not such a simple interaction of there's a probe binding to the aptamer and the target binds and the probe goes away, right? It's a little bit more complex than that, and even a bit more complex than what I'm showing you here, right? There's some kind of association between the probe and the aptamer, therefore there's a dissociation. There's also probably a K of folding for the aptamer to fold into a binding complement state, and then there's the actual interaction of the target with the aptamer. But none of our modeling could resolve why some of these weren't working, and so we just did what any good scientist should do, and we looked more carefully at the experiments. And so this is a zoomed in version of really reproducing what the papers did for these probes that we saw weren't working. And again, even when we followed the timing, we still weren't getting any interaction, but what we realized is that in these select publications, they were actually adding the aptamer to the target and then adding the probe or not, right? So it wasn't actually a displacement of the probe, it was simply if the aptamer is pre-folded and pre-bound to the target, then the probe couldn't interact. And so all my relief was there because I'm like, okay, we've reproduced these experiments. No one was lying. Aptamers are super robust. They feel discreet. But in reality, this isn't a great field testing biosensor, right? If you leave these to equilibrate over time, you're not going to have any response. So if you have one person doing the test within 5 minutes versus one person accidentally leaving it on the table for 2 hours, you're not going to get, oh, there's a contaminant in this food or not, right? So this is certainly not robust. And so we thought, okay, there must be a way to figure out what's going to make a good complementary probe thereafter. This should not be that hard of a problem, right? So we thought, okay, maybe we can just predict this by looking at simple properties. So we just used basically IDT DNA, like melting temperature prediction. And you can see And we're looking at the TM of the predicted TM in the buffer conditions we use on the x-axis. And y is a sort of a measure of how much switching or on biosensing you see. And you can see maybe that there is a pretty good correlation for generally what a good probe will result in biosensing. With very high TMs, obviously, you're never going to kick those off. But what you can hopefully see is that there's some yellow dots there and green dots there. The green ones represent biosensors that work pretty quickly within about 30 minutes, sometimes less, whereas the yellow dots take 2 hours to reach a response. So again, maybe not exactly the biosensors we want if we want to deploy them for rapid field testing. So while I was somewhat satisfied that, okay, TM maybe can help guide the design of probes, this was not enough. And if we think about all the amazing talks that we've heard about people using chemical modifications, I think that our TM predictions will break down pretty quickly. So then a very smart reviewer said, well, we know predicted TM is not that useful of a measure, so why don't you just measure the TMs like any good biophysical chemist? So we did that, and actually the interaction was, or the correlation was worse. We couldn't even measure P1 because it was such a low melting temperature, and otherwise the fits, we didn't get any better predictive data, so that was a sad story. Similarly, we thought, okay, maybe we're just not getting good enough fits to predict TM. Maybe we just need that first step of a biosensor, which is the association of the probe with the aptamer, because we know the aptamer can fold into these different structures. So maybe the actual binding affinity of the probe to the aptamer is what we need to look at. So for this, we just use a fluorescent polarization assay where we have the probe labeled, and when it's in solution, because the probe itself is very small, it rotates a lot, and therefore any excited polarized light generally becomes depolarized. But when the probe binds to the larger aptamer, then it rotates less quickly, and so the light remains a bit more polarized that's emitted. And so we did this for all the probes, and I'm not showing you any correlations here because we were just shocked by the fact that these so-called good biosensors that were like fast and effective had basically overlapping dissociation constants with terrible probes that almost didn't work at all or took hours and hours to work, or you needed to add this. In fact, a really good probe, P8 here, had the same dissociation constant as a probe that didn't work at all. So very disappointed that my limited and our lab's limited ability to use predictive measures to design new probes completely failed us. We thought, okay, maybe we need to do a rapid screening method, right? And so our group has really been very lucky to have surface plasmon resonance as a method in our lab. We certainly love other methods, but this is one that we can do quite quickly, and it's been useful for small molecules. And so the way this method works is with measuring a mass change at the surface, thanks to change in a refractive index that is responded to from this gold plasmon wave. And so we can actually measure in real time, of course, interactions at the surface, so either aptamer capture on our chip or binding in real time. And so we see increased responses when there's more at the chip, and we can see a decrease in response when things are coming off the chip. So this has worked for us really well. So we thought maybe we can use this to screen for probes that work. And so this is the way our sort of method works. So again, on the x-axis, we have time, and on the y-axis, we have that change in sort of a massive response at the surface. So we start off by having a very stable DNA DT oligo that we can keep on the chip for months, and we can do about 3,000 cycles. So really nice and robust for small bands. So then what we do is we capture our aptamer that we've extended with a poly A tail. Really cheap, really easy. We can do this with RNA, DNA. And so when we capture the aptamer to the surface, we see an increase in response because there's more at the surface. Then we go ahead and add our probe, maybe it causes a structural change or not. What we see, of course, is just another increase in response at the surface because we've added this large probe. Here we can also measure a little bit the dissociation constant of the probe itself, but for now we're just capturing it. Then finally we can add the target. And if the target causes the probe to dissociate, which is what you want in a biosensor, then we should actually see there we go, a decrease in signal because the small molecule is relatively small compared to these larger probe sequences. So the small molecule, even if it binds, is negligible compared to that mass change of the probe being released. And so that's the goal here. And we can use denaturing agents to basically rip everything off the chip and start brand new. And we can do this over and over again. And so we think this could be a really nice way to just rapidly and cheaply screen a bunch of probes where we're not limited to adding different fluorophore quenchers, etc. So let's see if it works. So this was the data I showed you earlier. So to kind of benchmark this, we picked a really good probe, P8, and a really bad probe, P2. And so you can see P2 in gray. Here we start with the probe is bound, and we make sure it stays stable. And then this is the point where we inject the small molecule. Again, it's a very small mass, so we don't really see much change. But with P2, basically the probe stays on, it doesn't get removed because it's not a displacement probe. Whereas P8, when we add the small molecule, it causes, I guess, a conformational change that kicks off that probe, and we can see that really nice curvature, that dissociation. And so we take just that phase after we've added the small molecule in, and then we model this with the decay. And we basically calculate the decay of the exponential decay to sort out which probe might be the best. And so we can look at this qualitatively, and you can imagine that these ones down here are better probes. The ones that are not dissociating are terrible probes. But we're also interested to see if there is a little bit more correlation we can do from there. And so we take that K from our SPR, but we compare it to that fluorescence data that we got. And here we decided to use sort of fluorescence recovery as our biosensor, let's say constant. And so this is basically how much turn on signal we get compared to when the probe is bound to the aptamer. And so when we do this simply with the things that give us a good K value, we actually get a really nice correlation. The data is limited, I recognize that, but this really showed some promise that this is not maybe just a screening tool, but actually something that we can use to at least semi-quantitatively or categorize and sort of organize different probes. So that was great, that's cool. But of course, these were all probes that were already published. So we wanted to know, could we use the system to design new probes and know if they were going to work or not? So trying to do things really incrementally, we just stuck with the same octotoxin aptamer. We just extended it on the 3' end a little bit. Basically looking at the original selection by Penner and just adding the same nucleobases from the whole length. And then we designed 2 probes in the exact same region, had very similar TMs, but just differed by base. And you can see from our SPR that we would probably expect P9 to be a good probe because it dissociates nicely from the surface, whereas P10 did not. And then when we did a fluorescence assay, of course, that's what we saw. So we're very happy with that. We then used our best probe and did a little biosensor that was able to allow us to detect octotoxinate at a very nice limit of detection that is often within the range that we need to do in field testing. So that was great, but can we move beyond the octotoxinate aptamer? So as a first step, I had students in my lab go through the literature and find other aptamers that people have done these displacement probes with. They tested about 6 and found that 3 had really nice displacement, right? So this idea where the aptamer is fluorescently labeled, there's some published complementary probe that causes quenching, and then when the target binds, the probe is released. So actually we got this data first, and then with those, we tested them on the SPR. And again, we see this nice decay, suggesting that this method actually works for other aptamers. It's great. So then, of course, the next incremental question is, okay, it does work for diverse aptamers, but can you really use this on an aptamer that hasn't been studied to death, like octotoxin? And so we chose to use this serotonin aptamer that was published in the functional guided development of aptamers that was published in Science by the Anna Anders group and Milan Stojanovic. And so at the time, there was only one other paper that had used their serotonin aptamer. And so we were like, oh, that's not quite, no one has ever touched it before. So then we did some minimization of it ourselves, just so that it would be different. And then we even made sure that we designed probes on the 3' end that no one had ever done before with any aptamer close to this. So we we're really convinced that, okay, hopefully we can justify that this is really something brand new. So we picked these probes because we now know approximately that we don't want to be too high on TF. And so we did our screen on SPR. You can see that only S5, probe 5, looked like it had any promise. And so again, we synthesized the aptamer, fluorescently labeled the probes with the quencher, and we did the assay. And again, you can see that the only probe by SPR S5 that showed any displacement is the one that worked with our fluorescence assay, whereas as a control, we did another one and we saw no biosensing. So we were really quite enthusiastic that this can help us sort of expedite our screening for developing biosensors, at least to small molecules. One thing we're still working on is if we can really apply it to proteins. With trigger warning, my SPR data here is terrible. So if you do SPR or BLI, please give me some, grades, I guess, when I show you that there's some bulk effects and mass transport limitations. But the problem with proteins, of course, is that proteins, when they bind, are going to have a huge mass change at the surface, and it's going to be very difficult to see that smaller probe release, right? Where that was really our benefit with the small molecules. But nevertheless, we tried to design some sequences based off the Yankee sort of paper here with thrombin, where we've extended the thrombin because that Aptamer is just so short. And we screened a couple different probes. And what we decided to pick for probes that we didn't see as much thrombin binding, our thought here being that thrombin is going to bind, it's going to cause a huge change in mass, but perhaps as the probe is being released, we'll see a slightly lower change in response. And so luckily, I think we were lucky that the ones that showed that behavior also allowed us to see some biosensing when we translated them to this fluorescent quencher biosensor. So that's just our brief little method. That was a huge research tangent on an ultimate goal to do single molecule studies. But we're still very happy with this method since we think that it is nice and fast. You don't have to order oligos that are complicated. They can be cheaply synthesized and screened. We're currently applying it to riboswitches, switches, so really showing that this is not DNA aptamer limited. We can do RNA aptamer, riboswitches. Switches. We really like that there's no dependence on the tag itself, because as you know, that different threat to fluorescent quenching, the distance matters here. And so we're not really looking at that property, we're focusing on the displacement aspect. And then because we've done it with different small molecules and RNA, we think there's nice broad applicability. We're also not limited if we want to start using modified backbones or even modified bases. And ultimately, while we do this in SPR, I think this method will work on any surface-based sort of platform. So if you use ELI or quartz crystal microbalance, or maybe some different cantilever studies, or certainly single molecule, we think that this could be applied to that as well. So with that, thank you for listening to our quick little method. Thanks to my lab who did all this great work, especially some undergrads who did the hard work finding some good aptamers and biosensors that work, and our collaborator who will be eventually doing the single molecule studies with Sabrina Westway, ABC, and funding. Thank you so much.
So, I think we've got time for a few questions. I'll open with one now. I'm just wondering, is there any association you saw between the selection technique used to make the aptamer and whether it's going to be successful as a biosensor?
That's a good question. I'd say at the moment, no, because ochratoxinate was kind of a classic selection method and it worked fine, but all the more recent ones are from Joanne Liu's lab at Waterloo, or the serotonin aptamer of the rational design, which used more of the capture sealant.